Hello, good afternoon. So I apologize for not being on the site this morning to conduct this lecture. This will be the last interaction. I think this is the last part of this really. Biotechnology applications in medicine. Start with the science fiction, not right. Tomatoes that fight cancer. Wouldn't that be nice? Peanuts that are hypoallergenic, potatoes that are pest resistant. Products of high alkaline salts. Yeah. So these realities of modern agriculture are a direct result of biotechnology. All right. Could read this background on your own time. I don't think we need to read a lot here. It's just a definition of biotechnology. It's defined as the application of scientific and technical advances in life science to develop commercial products in biotechnology. Biotechnology is a collection of technologies that capitalize on attributes of cells, such as their manufacturing capabilities, and put biological molecules such as DNA and proteins to work for us, like the last class where we discussed production of monoclonal antibodies. This is basically it, right? It fits into this definition. All right. Yeah. And of course, like I said earlier, biotechnology is a multidisciplinary test. You have the conglomeration of disciplines such as genetics, biomicrobiology, animal cell culture, molecular biology, biochemistry, embryology, cell biology, and even some engineering aspects. Biorobotics, chemical engineering, and so on. Information technology, especially the bioinformatics. We know. All right. The tools, of course. We're familiar with some of these tools: electrophoresis, DNA RNA purification, cell cultures, sequencing, microarray technology, which is popular in cancer research, bioinformatics, nanobiotech, stem cell technology, right? Animal models of human diseases. This is just a schematic chart showing the drug discovery process, research and developments, conventional compound screening and identification of lead. You need to find out what a lead and a hit compound is. Clinical studies where you use rat or mouse models. And that's where you also do your toxicity testing, although you have bioinformatic tools now that you use for toxicity. Due to the recent drive to promote animal ethics, if you can reduce the number of animals dying from toxicity experiments as much as you can, you should. So that's why in silico toxicity screening is replacing some of these conventional in vitro or in vivo toxicity tests we do. Clinical trials, phase 1, 2, and 3, and then safety, review and approval of a drug. So it takes a lot of years, do the plus or minus here. Looking at 10, 12 years, plus or minus, for a drug to either pass or fail to be approved and commercialized. All right. Okay. Blue biotechnology, marine and aquatic applications. The ocean is blue, right? The color of love. Green biotechnology, forest, the green shrubs. Yeah. Red biotech, medical biotech. The color of blood is red in it. Isn't it white biotechnology? It's industrial biotech. Okay, so we have in medicine, modern biotechnology is generally applied to areas such as biopharmaceutical production, pharmacogenomics, that's personalized medicine, genetic testing, genetic screening and gene therapy, predictive AI, artificial intelligence. This is the future of research. Okay. You can study this on your own. It's quite easy to understand. Nanoparticles, it's nanotechnology. Coating nanoparticles with drug to facilitate quicker delivery into your target cell. As application in a lot of diseases, including cancer, which is a nanoparticle drug delivery system. Ovarian cancer. Yeah. Biopharmaceuticals maybe use large bioreactors to produce drugs. Okay. All right. Some of the products that have come out from this technology include the human insulin. It's used for patients suffering from type 1 diabetes. You have other proteins which are listed here, including the monoclonal antibodies, which we've discussed in the last class. All right. Insulin, diabetes, right? What I just finished saying, you can check out the list. Okay. So biofarming as a term coined for the production of edible crops, wood containing biodrugs for use by human and animals, right? It's often parasitical. Your diet should also serve as medicine. Basically, that's what's happening here, right? Okay. Genetically engineered crops or fruits have been developed for anti-cancer drugs, vitamins, and essential antioxidant compounds, plus or minus antibiotics, and so on. Cosmeceuticals. Basically, biotechnology has infiltrated almost every aspect of life. Okay. I want to get the material. For example, a company in the US has made genetically engineered goods that produce 14 different kinds of therapeutic proteins in their milk. This is quite interesting. All right. And it takes about 18 months to produce such goods. For cows, it takes 3 years, but the milk yield is 10 times higher. All right. So the same thing. Yeah. Artificial cells, an age-old concept, much desired by researchers, biologists, and physicians, is now a possibility. Cell constructs are being used nowadays for blood cleansing, right? Activated chapel containing cells in uremia, poisonants, and liver failure. Future developments, hemoglobin delivery, enzyme replacement, drug delivery, gene therapy, and so on. Pharmacogenomics. This is quite interesting. Personalized medicine. This is the future of medicine. Pharmacogenomics refers to the use of genomic information to develop customized drugs. So the health system will cater to an individual, not a whole. Yeah. Pharmacogenetics is also used to describe the story of the ways genomic variations affect drug responses. Yeah. Pharmacogenomics take advantage of fact that individuals have unique genomes representing their genetic makeup. Each genome is likely to react differently to a particular drug and dose amount. Of course, it makes sense, doesn't it? Okay. What I just finished saying, in the future, physicians will be guided by each patient's unique genomic ID to prescribe drugs tailor-made for different genome types, right? We have a long way to go before this could be achieved, but we're going towards there and moving towards that their direction. And the SNP is nucleotide polymorphism. You should know the difference between a mutation and polymorphism, right? Yeah, proteomics, genomics, and so on. You have to take your time and read this uh, slide. Yeah. Gene silence. Regenerative medicine. This is where the stem cells will come into play. Induce pluripotent stem cells. We can't talk about regenerative medicine without talking about stem cells, bioinformatics, future uses of them. Right. Yeah. Yeah. I'm going to get the slide, so no worries. Your ability to read and understand on your own is part of a master's degree in it. So I wish you the best of luck. I'm going to send the instructions for an assignments to your class. After that, we'll talk about your continuous assessment as well. Thank you.